Hello students, welcome everyone for your online classes. So as you people know that we have started with the next semester, your even semesters. So we will be starting with the paper 7 of your 6th semester zoology. So you'll be having two papers, paper 7 and paper 8, as you have studied last year. I mean in the last sem, in 5th sem you have studied paper 5 and paper 6, and here you'll be studying paper 7 and paper 8, each for 100 marks. So this is 6.7 paper 7 applied zoology. So in this class I'll just brief you up the syllabus and the introduction of this all the syllabus which you will be studying in this whole semester. Okay, so here to tell you what is applied zoology. So what is the definition of the word applied zoology? It is a branch of zoology. Okay, it is one of the branch of the zoology which deals with the study of scientific knowledge and its application. It is a branch of zoology where the existing scientific knowledge is applied to develop more practical applications. Okay, what we'll do is scientific knowledge is applied, okay, and it is made to bring up many applications which are having benefits to the human welfare. Okay, so such a type of applications are called as and that branch is called as applied zoology. In this paper you will be having 10 units, okay, and you have 3 credits for this paper and it is for 45 hours. So here first unit 1 is introduction to host parasite relationship. Okay, so you can see there are 10 units and the time is also there, okay, and this is completely for 45 hours. So we'll see the unit 1. Here it is introduction to host parasite relationship. I think you people know what is host and what is parasite. So what are parasites? Parasites are the organisms which depends on other organism for their food and shelter. Okay, so these are the organisms which cannot have their own food and their own shelter. So ಅದೇನಾಗಿರುತ್ತೆ, ಬೇರೆ ಇನ್ನೊಂದು ಆರ್ಗನಿಸಮ್ ಮೇಲೆ ಡಿಪೆಂಡ್ ಆಗಿರುತ್ತೆ, and the organism on which these parasites are dependent are called as host. Okay, and some of the parasites are pathogens in the sense some of the parasites will cause us a disease. So in this unit it is for 2 hours, and here you'll be studying what is the definition of host and definitive host, then intermediate host. There are 2 types of host, definitive host and intermediate host. Okay, some of the parasites will have 2 hosts. For example, you have studied malarial parasites. If you have studied, I know here you'll be studying in detail in this particular paper. So that will be having what you can say 2 hosts, one is human and one is mosquito, like that. Intermediate host and definitive host. And then parasitism. What is parasitism? You know, that process of parasites, how they are depending on the host is called as parasitism. And then symbiosis is mutual association with that of the host. Okay, then commensalism, this is another type of association of host and parasites. And then reservoir, and then zoonosis. So these are the different type of host parasite relationship. Then so in unit 2, which is for 3 hours, you'll be studying epidemiology. What is epidemiology? This is the branch of medicine which deals with incidence, distribution, possible control of diseases and other factors related to the particular disease. Okay, so here you'll be studying the transmission, prevention and control of diseases. You'll be studying 2 diseases in this particular unit, tuberculosis and then typhoid. And then rickettsia and spirochetes. This is unit 3, which is for 4 hours. So what is rickettsia and spirochetes? These are the 2 different types of bacteria which lives on the organisms and which causes a severe disease, and some causes less severe diseases to human beings. Okay, what is rickettsia? Rickettsia is a gram-negative bacteria found in ticks, lice, fleas, mites and some of the mammals. Okay, it causes diseases to humans. Then spirochetes, these are the spiral shaped bacteria. Okay, as the name suggests, spirochetes, spiro means spiral shaped bacteria which causes diseases. Okay, these are also disease causing organisms like syphilis, relapsing fevers. These are the different diseases. Okay, and then Next, unit 4 is parasitic helminthes. Okay, you have studied helminths. There are two non-chordate phylums, okay, of helminths, platyhelminthes and aschelminthes. In platyhelminthes you have studied flatworms, and in aschelminthes you have studied roundworms. Okay, here you'll see some of the helminths which are parasites. You will study those. It is here you can study the life history of the helminths, and then pathogenicity. What is pathogenicity? It is the ability to cause a disease. What are pathogens? Pathogens are disease causing organisms. Here you'll study the life history and pathogenicity of three helminth, okay, three worms. First is Ancylostoma duodenale, and then Schistostoma hematobium, and then Ucheraria bancrofti. Okay, these are the three helminths. This is for 4 hours. And coming to unit 5, here you will be studying the applied aspect, that is sericulture. You know what is sericulture? It is the rearing of silkworms, okay, for the extraction of silk. It's a such as agro-based industry, okay, and then species of silkworm. You'll study mulberry and non-mulberry species, and then races of silkworm, and then brief account of mulberry cultivation. How will you cultivate the mulberry plants? You study that, and then life cycle of Bombyx murray. Bombyx murray is silkworm, and then structure and functions of the silk gland, then diseases of the silkworm. There are some of the diseases which are affected to the silkworms. There pebrine disease, then muscardine disease, then Grasseri disease, and Fletcheri disease. These are the some of the fungal, bacterial and viral diseases which are infected to the silkworms. And then byproducts of the silk industries. There are many byproducts which comes from the silk industry. You will study those byproducts. It is for 6 hours, and it is one of the largest chapter, large unit in this paper. And unit 6 is pisciculture. So pisciculture, it is the rearing or breeding of the fishes, okay, by artificial means. That is called as pisciculture, fish culture. In this you'll study pond culture, okay, different types of culture. Pond culture, then processing and preservation of fishes. How will you process and preserve the fish for long term? As if you know that many we used most of the fishes as a food fishes, okay. And then economic importance of fisheries, and then induced breeding. How will you induce the breeding? It is artificial means of inducing, what I can say, you are bringing the fishes into a breeding season by artificial means rather than natural one, okay. That process is called as hypophysation. And then principles and procedure of induced breeding. How will you go for this induced breeding? What are the principles and what are the procedures, and what are the different steps? And then advantages of induced breeding. Okay, induced breeding ಮಾಡಿರೋದನ್ನ ಅದರಿಂದ ನಿಮ್ಗೆ ಏನು ಅಡ್ವಾಂಟೇಜಸ್ ಇದೆ? ವಿ ಡೂ ಎವ್ರಿಥಿಂಗ್ ಫಾರ್ ಅ ರೀಸನ್. ದಟ್ಟು ನಮಗೆ ಹ್ಯೂಮನ್ಸ್ಗೆ ಅದರಿಂದ ಬೆನಿಫಿಟ್ ಆಗುತ್ತೆ ಅಂತ ವಿಲ್ ಡೆಫಿನೆಟ್ಲಿ ಗೋ ಫಾರ್ ದ್ಯಾಟ್, ಓಕೆ, ರಾದರ್ ದೆನ್ ಫಾರ್ ಎನಿ ಅದರ್ ಆನಿಮಲ್ ಆನ್ ದ ವರ್ಲ್ಡ್. There is something which is having a benefit for us, then we'll concentrate on that much, okay. And then coming to the unit 7, poultry. It is for 6 hours. Poultry, you know what is poultry? It is raising or rearing of the birds commercially or domestically for the purpose of meat, eggs, feathers, okay, etc. And here you can study the importance of egg in combating protein malnutrition. You know, egg is one of the protein rich diet, okay, which can be given to poor people who have malnutrition problem. And then poultry as subsidiary source of income. It can be used as a source of income as well. And then breeds of fowl. Fowl means cock and hen. There are two types of breeds: desi breeds, which is of our country, and then exotic breeds, which are of our different country, okay, from other country. And you can see 3 types of birds, I can say, hen and cock, and 3 types. In that, one is layer. These are the birds which can be used for laying eggs. Here broiler means these are the birds which can be used for meat purpose. Dual purpose means which can be used both for egg laying as well as for meat purpose, okay. And here you not only see the hen and the cock, you can see chicks, ducks, okay, goose, all these things. And then white leghorn, you'll study the different race, White Leghorn, which is called as Mediterranean class, and then Plymouth Rock, which is American class, and Sussex English class, Lexhan Asiatic breed, and then Giriraja. These are the different breeds. Then here you can also study the brief account of poultry farming. How will you go for poultry farming? What are the different procedures and what are the different techniques? Then diseases. Everywhere you have a disease, okay, as you have in sericulture and pisciculture, here also you'll have the disease, okay, which are being affected. Diseases of poultry fowls are Ranicate disease, fowl fox disease, avian leukosis, and coccidiosis. These are the different fungal, bacterial, viral, and protozoan diseases. And unit 8 is dairy. You know what is dairy? Here you can go for rearing of the cows for the purpose of milk and milk products as well as for meat. That is called as dairy. It is one of the branch of applied zoology. It's here you can study the composition of milk, nutritive value of milk, and then draft breed. You can study the different breeds of the cows and bulls here. Okay, it is also for 6 hours. You can study draft breed called as Hallikar, then milch breed called as Red Sindhi, and then dual purpose breed Ongol. Exotic means these are the foreign breeds called as Holstein Friesian, cross breed Red Den, and then buffalo breeds also you can see Nagpuri and Surti. And diseases in cattle, causes and effects of mastitis, anthrax, peticemia, foot and mouth disease, and brief account of the management of dairy farm. Okay, here also you can go for breeds. Breed can be used for only for the milk laying purpose or only for the meat purpose, okay, or for the dual purpose. Dual means for both the reasons, okay. And unit 9 is transgenic animals, which is for 5 hours. So what are transgenic animals? These are the animals that have foreign gene, okay, that is inserted into their genome. Okay, animals which are having a foreign gene inserted into their genome. You know what is genome? It is a sum total of all the genes present in an animal or a cell. Okay, here you can study introduction of transgenic animals and its advantage. Okay, there are many advantages here. Okay, maybe we can produce different varieties like disease resistant varieties, and then drought resistant varieties, okay, like this, and then giving more yield, high yielding varieties. We can produce all these good varieties from the transgenic purpose. Okay, here you can study nuclear transplantation method. What are the different types of or methods which is used to insert the gene into their genome? Okay, these are the methods used for gene insertion. First is nuclear transplantation method, which is used in dolly. Okay, you know that it is a sheep, which is the first transgenic animal. And then micro injection technique, which is used in mice. And then embryonic stem cell transplantation, which was used in chimeric mice. And then retroviral method, which is used in mice. Next, coming to the last unit, unit 10, assisted reproductive technologies. These are some of the techniques which is used to assist the process of reproduction. Andre normal way ಅಲ್ಲಿ ಆಗದೆ ಇರತಕ್ಕಂಥ reproductive process ನ ಇನ್ಕ್ರೀಸ್ ಮಾಡೋದಕ್ಕೆ ಆರ್ಟಿಫಿಷಿಯಲ್ ವೇ ಅಲ್ಲಿ ನಾವು ಅದನ್ನು ಇಂಪ್ರೂವ್ ಮಾಡೋದಕ್ಕೋಸ್ಕರ ಯೂಸ್ ಮಾಡುವಂಥ ಟೆಕ್ನಿಕ್ಸ್ನ ಅಥವಾ ಟೆಕ್ನಾಲಜಿನ ನಾವು ಅಸಿಸ್ಟೆಡ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಟೆಕ್ನಾಲಜಿ ಅಂತ ಕರಿತೀವಿ. ದಿಸ್ ಇಸ್ ಫಾರ್ 4 hours. This is here cryopreservation of semen. What is cryopreservation? Here you will preserve the semen in liquid nitrogen condition. Okay, minus degree centigrades ಅಲ್ಲಿ ನೀವು ಅದನ್ನು ಪ್ರಿಸರ್ವ್ ಮಾಡೋದು. Then you will study different techniques of assisted reproductive technology. First is AI technique, that is artificial insemination, and then GIFT technique, that is gamete intrafallopian transfer, and IVF, that is in vitro fertilization, and embryo transfer technique. ET means embryo transfer techniques. These are the different assisted reproductive technologies which can be used in your mostly in dairies, in poultry, in pisciculture and all. Okay, so this is about your unit, and these are the suggested readings. Okay, these are the publications which you can refer for your studies. Then coming to the practical paper, here it is for having 2 credits. Okay, and it is for 50 marks. Here you have for 15 units you'll be having 9 practicals here. So here you can see first question is identify and comment on following bacterial strains. Here you will study the following bacterial strains, where you can study Salmonella typhi, Microbacterium, Tuberculosis, Borrelia. Okay, these are the different strains. And then study of helminthes worms. You'll study some of the worms. And then morphology of Bombyx mori, that is silkworm. There you can study larva, pupa and moth. And then study of silk gland, that you can study with the help of photographs and pictures. And then identification and comment on food fishes of Karnataka, like Katla, Clarius, Labos, Acobranchus, etc. And then on breeds of fowl, we can use the photos. And estimation of protein in milk sample. And then lactometer test for water content in milk. And then project on dairy farming, breeds of cattle, then diseases of cattle, and pisciculture, poultry farming, transgenic animals, rearing of a silkworm. Okay, these are all the I can say questions which you have to complete in your practicals. And here you can see the scheme of practical examination for 50 marks, and which is for 3 hours. In first you have to identify and comment on bacterial strains, and in second you have to comment on helminth worms, and third morphology of silkworm you have to write, and then fourth on silk gland, fifth on food fishes, sixth question on breeds of fowl, and seventh question on estimation of protein in milk sample. It is by 2 method, titration or lactometer method. And then question 8 is subject project report submission, and 9 is class record. Okay, so this is about your practicals and your theory syllabus of paper 7 applied zoology, which is first paper in 6th semester. Okay, with this I will be stopping, and in the next sessions I'll be starting with the proper syllabus. Okay, so thank you everyone.